Welcome to the podcast on Dialogues in Science and Technology, PDST, where lab breakthroughs meet everyday life. Today we're diving into astonishingly amazing world of circular RNAs, tiny ring-shaped molecules, once written off as noise, now recognized as powerful gene regulators, biomarkers, and potential therapies. In this episode, we'll unpack what makes circRNAs so unique. Hint, think about their loop. You guessed it right, the loops or closed circles makes them super stable. We will see how they're linked to various functions like brain health, cancer, and aging, and why they could reshape diagnostics and drug design. Grab your curiosity. Let's make the science and technology conversational.
Imagine finding out something you thought was just, cellular junk. Actually turns out to be a key player, like finding a hidden gem.
It happens more than you'd think in science. And today, we're diving into one of those stories, circular RNAs or circRNAs.
Right, things scientists used to dismiss back in the 70s as just transcriptional noise.
Exactly, just splicing errors maybe. But fast forward, and wow, the picture's totally different.
So that's our mission today, isn't it? To really unpack what these circRNAs are, especially this idea of them being microRNA sponges. We want to get into why that matters for, how your cells work and what happens in disease.
Absolutely. We'll look at their stability, how they interact, and even touch on some of the debates about how big their impact really is.
Okay, so where did this rediscovery start? How do we go from noise to, something important?
It really came down to new technology. Things like RNA sequencing let us see everything that's being transcribed in a cell, not just the usual suspects.
And suddenly, these circular molecules popped up everywhere.
Everywhere. And not just randomly, they turned out to be abundant, really diverse, and conserved across different species. That challenged the whole artifact idea pretty strongly.
So what makes them circular? What's the structure?
Okay, so unlike typical RNA, which is linear like a piece of string, these are actual loops, covalently closed loops.
How do they form that loop?
Through a neat process called back-splicing. Basically, instead of joining sequential bits of the RNA transcript, an end part, a splice donor site, loops back and connects to an earlier part, an acceptor site. Boom, you get a circle.
And that closed loop structure, that's the key to their stability, right?
That's a huge part of it. Normal linear RNAs have exposed ends, a 5-foot cap and a 3-foot poly-A tail, which are like eat-me signals for certain enzymes, exonucleases.
But circRNAs don't have those.
Nope. Being closed loops, they lack those ends. So these enzymes, like RNAs they just can't easily latch on and chew them up.
So they last much longer.
Much, much longer. We're talking half-life sometimes over 48 hours compared to maybe 10 hours for a typical linear messenger RNA. Incredible persistence.
And that persistence lets them do things other RNAs can't, like being these sponges.
Exactly. Think of it, they hang around acting like these durable, reusable molecular mops or sinks, ready to bind things.
Okay, so if they're the sponge, what are they soaking up?
Primarily microRNAs, miRNAs.
Ah, right. The tiny RNAs that regulate gene expression.
Yeah, these little 22 nucleotide regulators. They usually bind to messenger RNAs, mRNAs, often in the 3' untranslated region.
And that tells the cell, okay, either stop making protein from this mRNA or just degrade the mRNA entirely. They fine-tune things.
Precisely. They're part of this complex called RIC, the RNA-induced silencing complex. They guide RISC to the target mRNA for silencing.
So how does the circRNA sponge interfere with that?
It's based on this idea called the competing endogenous RNA hypothesis, the CERNA hypothesis.
Okay.
The idea is there's a limited pool of each type of miRNA in the cell. And lots of different RNA molecules, mRNAs, long non-coding RNAs, and circRNAs have binding sites for these miRNAs. They all compete.
Like musical chairs for miRNAs.
Kind of, yeah. And circRNAs can be really good competitors because they often have multiple binding sites, these miRNA response elements, or MREs, packed onto one molecule.
So they just grab onto the miRNAs?
They bind them, effectively sequestering them, pulling them out of circulation. They sponge them up.
And if the miRNAs are stuck to the circRNA?
They can't bind to their usual mRNA targets, so the repression on those mRNA is lifted. They get de-repressed, meaning more protein can be made from them.
Ah, I see. It's a chain reaction. circRNA grabs miRNA, miRNA lets go of mRNA, mRNA makes more protein.
You got it. The circRNA, miRNA, mRNA axis. And what's clever is how they bind. Often the binding isn't perfectly complementary.
Why is that important?
Because perfect complementarity often leads to the miRNA guiding RISC to cleave the RNA it's bound to.
Yeah.
The imperfect binding lets the circRNA hold on to the miRNA without getting destroyed itself. It can keep sponging.
That is clever. Are there any famous examples of these sponges?
Oh, definitely. The poster child is SURO-S7, also called CDR1As. It's incredible. It has something like 70 binding sites for one specific microRNA, miR7.
70, wow.
Yeah, it's a super sponge. It mocks up miR7 very effectively, which is important in the brain for neuron function development. But it's also often messed up in cancers because miR7 can act as a tumor suppressor.
So SURO-S7 removes the suppressor.
It can, yes. Other examples exist too, like circStRI found early on in the testes, sponging miR138, or circPK3, which sponges several different miRNAs.
Okay, so the mechanism is definitely real, at least for these super sponges. But I remember reading there's some debate about how common this is.
That's a really important point, the stoichiometry challenge. The issue is that most circRNAs are actually present at pretty low levels in the cell, much lower than many mRNAs.
So numerically, it's hard for them to make a big dent in the miRNA pool.
Exactly. Can a few circRNA molecules really sponge up enough miRNAs to significantly de-repress lots of target mRNAs? It's a numbers game. Plus, some early studies used artificial systems, like massively overexpressing a circRNA, which might not reflect what happens naturally.
So the jury's still out on whether every circRNA is a major sponge.
Right. Sponging is one validated function, especially for abundant ones like SURS7, but it's probably not the only function or even the main function for many circRNAs. They can bind proteins, regulate their parent gene, maybe even get translated sometimes. It's complex.
Which brings us to medicine. If these are stable and have these functions, what's the potential?
Huge potential. Because they're so stable, especially in body fluids like blood or saliva, they're really promising as biomarkers. Think non-invasive liquid biopsies to detect or monitor diseases like cancer, heart disease, neurodegenerative disorders.
That stability is a real advantage there. And what about using them as drugs, as therapeutics?
Also very exciting. You could engineer synthetic circRNAs designed to sponge up specific miRNAs that are driving a disease. Or, because they last longer and might be less likely to trigger an immune reaction, people are exploring them for vaccines or as platforms to deliver instructions for making therapeutic proteins.
Using things like those lipid nanoparticles to deliver them.
Exactly. LNPs are key for delivery. It's a very active area of research.
So quite a journey. From overlooked cellular noise to these potentially powerful regulators and tools.
It really is. It shows how much we're still learning about the basic wiring of our cells and the importance of rigorous science to figure out what's truly significant.
Definitely makes you think, doesn't it? Considering circRNAs were hiding in plain sight for so long, what other seemingly minor biological bits and pieces are lurking in your cells right now, just waiting for us to realize they hold the key to something big?
Thanks for listening to the podcast on Dialogues in Science and Technology, PDST, and our deep dive into circular RNA. That's all for today's circRNA Spotlight. Stay curious, and see you next time.